Hi everyone, this is a final exam review for Chem 255, Introduction to Biochemistry at St. Francis Xavier University. I want to reiterate as I start the main themes that you guys should be looking at in studying for this exam. So some things that we come back to fairly frequently that are important in understanding a lot of the concepts that might bridge some of the concepts in the second half are the idea that proteins change shape to accomplish a certain goal. Anything that we've returned to a few times. So connections between metabolic pathways are fairly important. The intermediates at those connection points are also fairly important. We saw that some enzymes and some pathways will use certain strategies to accomplish a certain goal. Some of those strategies are used multiple times because they exploit a certain characteristic that allows a reaction to move forward. So things like decarboxylation or the use of UDP or pyrophosphate are things that might be important to know for the final exam. Understand too how pathways are directed. We talked about some basic regulation and didn't get into hormonal regulation too much, but just inside the cell, the signals that will drive a pathway forward or backwards usually relate to energy charge or substrates and products. So things like that are common in a lot of the pathways that we've talked about already in the second half of the course. I'll give you some information on the final exam as well, just how it will be set up. It will be similar to the midterm in that it will be split between multiple choice and short answer questions. I'll keep the number of multiple choice similar to what was on the midterm, maybe a couple more, but it won't be scaled up with the time that we have. We have 2.5 hours versus 50 minutes for the midterm. So overall, as a percentage, the multiple choice will be a little bit of a smaller percentage. I do want to add a few more multiple choice, a few more short answer. I'm looking at maybe 3 or 4 more short answer. They'll be in the 3 to 5 mark range, 3 to 6 mark range, something like that. And the number of points generally reflects the pieces of information that you should give in each of those cases. The material that the exam will cover will largely be from the second half of the course. Now, I mentioned this already in class, but I get a lot of questions about it still. It will largely be focused on the concepts, the pathways, the enzymes in the second half of the course. But you'll need to understand the information from the first half in order to describe, to explain the phenomena that we saw in the second half. A lot of the second half focused on proteins. If you think about it, proteins are the infrastructure for metabolism. We started our discussion with some non-enzymatic, things that you might call non-enzymatic proteins, hemoglobin and myoglobin. Even though they do catalyze some enzymatic reactions, these are largely transport proteins for oxygen. And so I'll start my discussion, my review here, before we get into enzymes and pathways and putting the bricks together in the metabolism building. These transport proteins, hemoglobin and myoglobin, underscore the idea that conformational changes are important. When oxygen binds to hemoglobin or myoglobin, the porphyrin ring system, heme as a whole, changes shape. It wants to sit flat, but when oxygen is not bound, the bonds between the iron molecule and the porphyrin ring are just too long to let that happen. And so once oxygen binds, it grabs hold of the electrons in the iron atom. It pulls them away from the porphyrin ring and allows the bonds to shorten slightly and that to the heme molecule to sit flat. Now this movement is transmitted through a histidine group that is attached to that iron molecule. And the chain reaction ends up being that the entire protein shifts. It changes shape slightly. And this has some important ramifications for the ability of hemoglobin to bind oxygen. Not only that, but this shifting, this chain reaction is carried through other subunits in the protein. So hemoglobin is a multi-subunit protein. There's 4 different subunits. Each one binds oxygen. And if one binds oxygen, this change in shape will make it easier for the other subunits to bind as well. So conformational change is a large part of why oxygen is able to bind hemoglobin and why all subunits in hemoglobin are able to bind oxygen. There are some other factors that we discussed which affect the ability of hemoglobin to bind oxygen. One of them was the Bohr effect. The Bohr effect simply explained that with a lower pH or a higher acidity, hemoglobin would release more oxygen. This mechanism is driven again by a conformational change. With more protons, something happens to push hemoglobin into its T state, which is the state that doesn't like to bind oxygen, and helps it release oxygen. It doesn't change so much the ability to load oxygen onto hemoglobin. What's really important here is that a decrease in pH will push more oxygen off of hemoglobin. And you can see that clearly here, where at any given oxygen concentration in the blood or in the body, with a lower pH, hemoglobin will contain less oxygen, will bind less oxygen. And this is important for tissues at the periphery or situations where there might be an increase in acidity. Generally, that signals that we need more oxygen. An increase in acidity isn't the only factor that signals a need for more oxygen. A slight decrease in oxygen at the periphery, certainly in the muscle when it is active, it uses oxygen. At the end of aerobic respiration, it reduces oxygen to water, and so this increases the gradient. Oxygen will travel downhill from hemoglobin into the tissues because of this gradient. CO2 is a byproduct of metabolism, and it needs to be carried to the lungs in order to be expelled from the body. This is accomplished in a few ways. We saw how CO2 can bind to hemoglobin. CO2 can be converted into bicarbonate and some hydrogen ions through the action of carbonic anhydrase. And while this helps bind and transport CO2, it also has the added benefit of increasing acidity and encouraging the release of oxygen from hemoglobin at the tissues where CO2 is high. Some other factors like temperature are also at play, or the production of metabolic byproducts like 2,3-BPG. These things all will encourage release of oxygen in a situation or a location where they're all present. Then the reverse happens when those stimuli are abated, when they're gone, or when there's less of an influence of those stimuli. So at the lungs, the gradients for oxygen and hemoglobin are reversed. It tends to move those two gases in the opposite directions. Also, acidity is presumably lower. There's less of an influence of metabolic byproducts like 2,3-BPG. And temperature might also be lower, although perhaps not as much as we might think. The main goal, though, is that the drivers work in the opposite direction to offload CO2 and load oxygen back onto deoxygenated hemoglobin. So it's important to understand how all those myriad of factors work together to accomplish this goal. Hemoglobin and myoglobin were really our only stopover on our discussion of non-enzymatic proteins, quote unquote. The rest of the time we spent discussing enzymatic proteins that are involved and critically important in metabolic pathways, metabolic processes. These enzymes are simply catalysts, things that make reactions happen faster. It doesn't make them happen, period. It just makes them more likely. Reactions can still happen without enzymes, but catalysts will greatly accelerate the ability of that reaction to move forward. We've seen mostly that these catalysts remain unchanged, and the method that they use to facilitate reactions is specifically to reduce the activation energy that's required in these reactions. So we say that enzymes catalyze reactions. Enzymes are generally specific to one role or one reaction. They are complementary usually to one substrate. They're stereospecific for one isoform, either the D or the L isoform. and they are shaped to accommodate a substrate of another specific shape. They also tend to have electronic complementarity, which is simply the characteristics of the amino acids inside the active site that match up with the complementary characteristics of the substrate. So hydrophobic amino acids like to be linked together, positive charge attracts negative charge, and these characteristics ensure that enzymes only target specific substrates. This is good because enzymes usually only mediate specific reactions, and if we link these together in a pathway, we want to make sure that it is directed towards one common goal. Here we see an example of a static protein, but there are also some instances where proteins will change shape slightly in response to a given stimuli to accept their substrates. This is what's called induced fit, and usually this is brought on by changes in the environment, possibly by a kinase that might phosphorylate an enzyme, or it might respond to a change in the energy charge of the cell or a change in acidity in the cell. A slight conformational change which allows access of the substrate to the active site. This is what we call induced fit, and it goes against the point on the last slide that enzymes are unchanged in the process, but normally this is reversed after the stimulus goes away or after the reaction is carried out. So the net effect is that they are still unchanged. Here we see a depiction of what an enzyme does to Gibbs standard free energy, or Gibbs free energy. Notice delta G is simply the difference between the energy level of the substrates and products, and it is the same regardless of whether the reaction is catalyzed or not. What the catalyst does is decrease the activation energy that's required to perform the reaction. The activation energy is shown by the hump in the middle, and so in an uncatalyzed state, a lot of energy is required to input into this reaction in order for it to move forward. What we see, the catalyzed reaction, where the main difference is that there's less activation energy required. We affect the enzyme's ability to mediate a reaction by controlling its activity. A large part of this is by inhibiting the enzyme. We can also activate the enzyme, and we talked about a few ways that we can do that, but enzyme inhibition is one major way that we direct flow of intermediates in a pathway, that we can slow down a reaction if we don't need it, and this is shown here. We talked about how most reactions function according to Michaelis-Menten kinetics, and that's indicated by the green line, where as we increase the amount of substrates that are present that can bind to and be acted on by the enzyme, that the rate of that reaction increases. Now this increases in a hyperbolic fashion. There is a point where you will saturate the enzyme. It can't work on any more substrates. This point is called Vmax, and there's no extra addition of substrates that can move you above Vmax in most situations. Km along the bottom is a measure of how sensitive the enzyme is to its substrates. How quickly does it get to Vmax? So if Km is small, that indicates that the enzyme is quite sensitive and it reaches Vmax quickly. What we see with competitive inhibition, this is where some other intermediate, some other molecule is competing for the active site and pushing the substrate out of the way. With competitive inhibition, the sensitivity seems to go down. That's what's indicated here in these blue and purple lines. A is simply, or alpha is simply a measure of how much of the competitive inhibitor is present. And so you can imagine with more competitive inhibitor, there's more of a resistance for the substrate to bind to the active site. The enzyme seems to react in a less sensitive way, but importantly, if you increased the substrate concentration high enough, you could still reach Vmax. It would just take a lot more substrate. This is different from uncompetitive inhibition, where no matter how much substrate you have, you can't reach Vmax. Uncompetitive inhibition is caused when some other molecule binds to a site that is not the active site on an enzyme and changes the shape of the active site. So no matter how much substrate you have, it doesn't bind as well to the active site. It won't reach Vmax. It's less sensitive as an enzyme for its substrate, and this will be maintained until that stimulus leaves, until the enzyme is returned to its normal shape. So knowing the characteristics, the general characteristics of enzymes, we set out to put them into context, to explore how the body uses these enzymes, links reactions together to accomplish a goal. And the first pathway that we discussed, the goal of which is to break down carbohydrates into a usable form to make energy, is glycolysis. Now, glycolysis is a series of 10 reactions. We focused on a few of those. We didn't highlight all of them. We focused on the ones that seemed to be important in allowing this process to move forward. Generally things where ATP is consumed or produced, or where high energy compounds such as NADH are consumed or produced, or any situation where something unique happens. These are the reactions that we've started to look at a bit more closely. Some of them shown here are hexokinase. This is the entry point, the first committed step to glycolysis. Conformational change is an important player here in the reaction of this enzyme. Phosphofructokinase, a second reaction, also uses an ATP, and it's the regulated enzyme that controls flow in glycolysis. These are what we consider the investment steps, because ATP is used in order to process glucose. We see aldolase, the splitting step, which is critically important, and it's by nature of splitting glucose in two that we get a return on our ATP investment, and we see that in the second phase. This is also the first place where we were introduced to glyceraldehyde 3-phosphate and dihydroxyacetone phosphate, intermediates that seem to pop up a few times, certainly in triacylglyceride synthesis and in some other pathways that connect to glycolysis. The second stage of glycolysis, we focused on three other main enzymes for various reasons. GAPDH is an enzyme that produces an NADH, so it's a high energy compound that can be used to make ATP. We saw later in the course that for every NADH, we can effectively assume that 2.5 ATP are produced if we completely oxidize the NADH. Keep in mind here that this is all occurring in the cytosol, and we will need to move NADH into the mitochondria to be able to gain those 2.5 ATP. And so some considerations have to be made in order to move this into the mitochondria. We didn't see these specifically in this course, which is unfortunate, but it's something just to still keep in mind while you're thinking about these different metabolic pathways. Now, oxidative phosphorylation will use pyruvate if it's present and if it has the capacity to do so. But in situations where there's too much pyruvate accumulating, in humans we can reduce that to lactate. Yeast have another mechanism of fermentation that produces ethanol, which we don't have. And this is related to the phenomenon of lactic acid that we see in the general population, or that is an idea propagated in the general population, where when a tissue is anaerobic, we produce lactic acid, which makes the muscle acidic and then is partially responsible for muscle soreness and the inability to continue exercising. But the truth is that muscles or tissues are never truly anaerobic without oxygen, but they employ non-oxygen requiring reactions when energy demand is high. So oxidative phosphorylation can only work so fast if the demand is very high, that pyruvate is accumulating in the cell. We'll use a non-oxygen requiring reaction, fermentation to lactate, to help produce more energy when it's needed. And we discussed at length what actual phenomenon are responsible for the production of metabolically generated acid in the muscle when energy demand is high. First of all, ATP breakdown itself releases protons, as shown here in this diagram. Not only that, but it seems that glycolysis is the acid-producing pathway and not removal of pyruvate to lactate. This reaction actually acts to buffer protons inside the cell. But in a situation where glycolytic flux is higher than removal by lactate, essentially this reaction will remove less of the acidity, remove fewer protons, and result in the accumulation of protons in the cell, the accumulation or the increase in acidity. Now, glycolysis is certainly at the center of our carbohydrate universe, but we have the ability to also metabolize other hexoses. In some situations, they're quite straightforward, a few simple reactions that allow these other carbohydrates to enter glycolysis. In some situations, they're a bit more complex, and in some situations, they're dependent on the tissue that you're in. For instance, fructose is one that we revisited a few times. It's fairly direct in tissues like the muscle, where hexokinase is a forgiving enzyme that can work on many different hexoses. It enters glycolysis in one single step. But in the liver, which doesn't have hexokinase, it can't enter glycolysis in one step. It's still phosphorylated by fructokinase, but the phosphate is attached at a different carbon, which requires necessarily that it is modified before it can enter glycolysis. And in modifying fructose, we produce a few different intermediates, some that we've seen before. Glycerol 3-phosphate is one that we see later in the course that is used to synthesize the triglyceride backbone. We produce dihydroxyacetone phosphate, glyceraldehyde 3-phosphate. These are intermediates in glycolysis that act to connect these two pathways. It's a point of connection with other pathways as well. And importantly, in the liver, this process bypasses phosphofructokinase. The step that tries to regulate flux and glycolysis is bypassed, so any addition of fructose will be metabolized to pyruvate and tried to enter into the TCA cycle. This has ramifications for excessive consumption of glucose and how it is stored in the body. We also mentioned that carbohydrates in the body can be stored for later use and then mobilized afterwards. Carbohydrates are stored as glycogen in the muscle and the liver and also somewhat in the brain. This is done by funneling off G6P from glycolysis and then linking them together in this branched glycogen granule, we call them. We funnel off G6P, and the synthesis and degradation of glycogen was largely self-directed in this course. There are a couple really interesting mechanisms that these enzymes use to accomplish these goals. For instance, the breakdown of glycogen. After it's been stored in this branched chain molecule, glycogen phosphorylase initiates a simple acid-base catalysis by a vitamin B6 derivative. This is PLP, pyridoxal 5-phosphate. And simply by donating a proton through an inorganic phosphate, we can break off one glucose from the end of a glycogen branch. And then in doing so, we create this negatively charged oxonium ion that attracts the phosphate that was used to cleave the glucose, and then in turn restores the proton back to PLP. So it's this acid catalysis that is quickly reversed in order to cleave glucose. We also saw that once glucose had been stored by glycogen, if we then mobilize it, we bypass one ATP consuming step in glycolysis. So in essence, we only invest one more ATP when we break down glucose from glycogen than if we were to break down glucose from blood glucose, or not from glycogen. This is attractive in the muscle for situations when we need a lot of energy. And it's why glycogen breakdown is such a fast process and why phosphorylase can respond so quickly to changes in the energy state in the cell. Carbohydrate metabolism wasn't the only section in the second half of the course. We talked about our other main energy-yielding macronutrient, which is fat. We talked about a similar process where we need to activate fat, transport fat, and then oxidize fat, much like glucose has to be activated, it has to be metabolized, and then oxidized. We activate fat through an ATP-linked pyrophosphate breakdown. We have to transport it in a unique way. We use CoA, coenzyme A, as a way to label fats that are intended for breakdown. And then once we have them inside the mitochondria, we can oxidize them through this repeated cycle of beta oxidation, where we produce reducing equivalents, and then we chop off small 2-carbon pieces that end up being acetyl-CoA, which are directed to the TCA cycle. We've since seen that for each one of these, we produce 3 NADH, 1 FADH2, and 1 GTP, which is the equivalent of ATP, allowing a 16-carbon palmitate to generate a net 106 ATP after we expend energy to activate it and transport it, etc. So lots of potential to produce energy from fat stores. Very energetic, energy-dense food source. The way that we transport fatty acids after activation, now we've skipped over activation. Notice over here where we have the CoA-bound fatty acid. This is our activated fatty acid. We've done the ATP-linked activation. We can't move CoA across the membrane though because it's simply too large. And so we bind it to carnitine, which is specifically exchanged with free carnitine across the inner mitochondrial membrane and into the mitochondria. Once we get it inside, we restore the CoA. We swap out CoA for carnitine. This is a thioester substitution, so energetically neutral. And notice the CoA compartments are separated here. CoA doesn't travel across the membrane. We have intact pools inside and outside the mitochondria. This linked CoA then undergoes beta oxidation, which are rounds of oxidation, hydration, oxidation, and then finally thioester cleavage. We saw a schematic of how one of these enzymes works. This is the enzyme that produces FADH2. It's the first oxidation in the pathway, and it's actually connected to the electron transport chain. This protein is part of that chain. And we can see in this diagram how the fat and the FADH2 are positioned so that the beta carbon, indicated here by this white circle, can specifically be oxidized. Now ultimately, the breakdown product of beta oxidation is acetyl-CoA. And this is a fundamental subunit for the TCA cycle. It's the subunit that both carbohydrate and fat are broken down into. Metabolism of these two fuels produces acetyl-CoA. And acetyl-CoA is also an important constituent precursor for fatty acid synthesis. The first committed step to make fatty acids is to carboxylate acetyl-CoA. And by doing that, we make the immediate precursor for fatty acid synthesis called malonyl-CoA. Acetyl-CoA is the source of malonyl-CoA though, so it's still an important constituent substrate for this process. Anytime we have acetyl-CoA building up that we're not using to make energy, we'll store in fat in this way by carboxylating it to make malonyl-CoA. This, importantly, again, we have to consider the context and the localization of these reactions. Remember, acetyl-CoA is largely produced by the breakdown of energetic molecules and organic compounds inside the mitochondria, but this reaction occurs outside in the cytosol. And so we have to have a way to get the acetyl group from CoA into the cytosol where we can resynthesize acetyl-CoA, because remember, CoA can't move across the membrane, and then we make this molecule which can be linked together to make fatty acid chains. These malonyl-CoA molecules are linked together by fatty acid synthase, a really fascinating protein that creates these two metabolic chambers that include all of the required enzymatic active sites in close proximity to the growing fatty acid chain. We bind malonyl-CoA first to ACP. This is our linker, or our flag molecule. It's analogous to CoA, but is used for synthesis. And ACP holds on to this growing fatty acid chain and then visits each of the enzymes in turn in these reaction chambers to add, to reduce, and to dehydrate and condense this fatty acid chain. We talked about the enzymes that are required to do this. There's two subunits and two full complements of these enzymes on either side. The lower portion of each side tends to perform the condensation reactions, where we link malonyl-CoA and we link acetyl-CoA to ACP. We substitute CoA for ACP. And then we link them together. Remember importantly that the acetyl ACP must be the seed to grow a fatty acid. Malonyl ACP is the building block that is added on top of that and is condensed together to form the fatty acid, and this happens in the lower half of the protein. The upper half, when ACP transmits or visits these other enzymes with the attached fatty acid, is what performs the beta-keto reductions and the dehydrations, ensuring that the fat is saturated and that ultimately we produce 16-carbon palmitate. We also spent a fair bit of time discussing how these two major catabolic pathways can merge. They both ultimately produce acetyl-CoA, and in a situation where acetyl-CoA builds up, we see a phenomenon which is more common in today's society where carbohydrates can be stored as fat simply because these two pathways can connect at the acetyl-CoA step. So generally when there's low metabolic rate and acetyl-CoA builds up, tissues will direct it to fat synthesis. Typically the liver and adipose are tissues that will do this. If we have excess glucose, the muscle tries to get rid of it. It is responsible for a large portion of glucose disposal, but if it's full, something else has to take up this role. And especially in the liver, when we highlight some carbohydrate molecules and how they enter glycolysis, we try to turn down glycolytic flux and prevent glucose from being metabolized to acetyl-CoA. But excessive fructose intake can circumvent that regulatory step, and it must be metabolized at that point. It will produce acetyl-CoA, and because it's not being used for energy, it's diverted to fatty acid synthesis. It's transported out of the cell and then is used to make new fatty acids. The point of connection between these two catabolic pathways is the PDH complex. It is one of the biggest complexes that we know of, one of the most tightly regulated complexes that we know of, and most important because it's the entry point. It's the nexus between carbohydrate metabolism in the cytosol and then the production of acetyl-CoA in the mitochondria. It's tightly regulated for that reason because it bridges these two realms. And this complex, made of 3 different enzymes, catalyzes 5 reactions. In general, the reactions are listed here. We need to get the acetyl group from pyruvate and position it in such a way that we can bind it to CoA. In doing this, we reduce some constituents of the complex, and they must be reoxidized. In reoxidizing those parts of the complex, we produce NADH, which we can also use for energy further on in metabolism. And the localization of these reactions is intricate and fairly important in this context, certainly. Enzyme 1 and enzyme 3 we talked about in the outer shell, and enzyme 2 at the core need to be bridged. They need some form of communication for these reactions to move forward. So pyruvate as a substrate binds in the shell. It binds to enzyme 1. Enzyme 1 then releases the acetyl group, and I'm saying that with an inflection because when it's bound to TPP at enzyme 1, it's not technically called acetyl group. It releases the acetyl group to enzyme 2. Here it now is an acetyl group. Enzyme 2 is what transfers this intermediate between these two phases, between the shell and between the core. Enzyme 2 is reduced in the process. It transfers a thioester transfer 2-CoA, which is energetically neutral, at the core, inside the core of this complex, and then it needs to be reoxidized. The oxidation occurs back in the shell of this complex, and it's mediated by enzyme 3. So acetyl transfer at the core, we recharge the lipoamide arm at the shell of the complex, and then we can redo this cycle all over again. We see this highlighted here, where the shell of the complex contains enzymes 1 and 2, enzymes 1 and 3. Enzyme 2 is in the center, and then appendages of enzyme 2 project outwards from the core to the shell, and then we have these mobile arms with the cup-shaped appendages, which are the lipoamide section of enzyme 2. So we can see here how substrate binding at the shell needs to be linked to acetyl transfer at the core. This happens by the movement of the lipoamide arm, and then in the process the lipoamide arm is reduced. It needs to be reoxidized, and by transitioning, by moving back to the core, it's accomplished, and then it can pick up another acetyl group from pyruvate and start the whole cycle all over again. It's only after this point that carbohydrate metabolism, the organic remnants of carbohydrate metabolism, enter the TCA cycle. Acetyl-CoA is the point of entry into the TCA cycle. We see how acetyl-CoA enters the TCA cycle here, and this is a fairly complicated diagram, but first we'll just highlight that from pyruvate to acetyl-CoA, this entire complicated step indicated here by one small black arrow produces acetyl-CoA, which can combine with oxaloacetate to enter the TCA cycle. The TCA cycle itself we consider the hub of metabolism simply because it has so many connections to other pathways, and we've already discussed these. Some of them, such as oxaloacetate producing malate and using this intermediate for gluconeogenesis, one that we've talked about in the carbohydrate section already. We've talked about in the fatty acid section how odd chain fatty acids are input into the TCA cycle directly by producing succinyl-CoA. And then there are a whole host of entry and exit points for amino acids, which we didn't get to cover in this course, but which allow the synthesis or the addition of organic compounds to the cycle depending on the needs of the cell at large. Certainly, if processes remove these intermediates, we won't be able to continue action of the TCA cycle. We need intermediates in order to inject acetyl-CoA into the cycle. If we remove the intermediates, we can't do that. And so one way around that is we carboxylate pyruvate to increase the intermediates in the cycle. This is called anaplerosis. This occurs because acetyl-CoA might build up if it's not being taken up and directed to oxidation in the TCA cycle. So some really unique regulation here by substrates and products that allows the TCA cycle to continue functioning. Now overall, the second half of the course has been very involved, and certainly much more involved than we've discussed here. There is a lot more detail, there's a lot more connection and regulation and importance of certain proteins that we haven't had the time to go through in this lecture. And I really encourage you not to use this as your sole method of studying. You have to review the lectures. You have to substantiate the lectures by information in the book and the assigned readings. There's a lot more to cover and a lot more in integrating these concepts than we've discussed here. We simply don't have the time for it. So I hope this has been helpful. And as always, if you have questions, please shoot me an email, and I'd be happy to get back to you and help you out with those. A lot of you are taking advantage of that already, and so I apologize if my responses aren't immediate, but I will get to all your questions. So all the best, good luck studying, and I'll see you at the exam.